Let's start the phylum Porifera. Porous, fera, bearing. These are the pore bearing animals. They are commonly called as sponges. Actually, the poriferans are the first metazoans, earliest metazoans. They are generally marine, means some are fresh waters, but mostly they are seen in the marine waters, in the oceans and in the seas. And the body is mostly asymmetrical body. Already we come across in the previous session that asymmetrical body is the one, if you go through any plane, is not possible of obtaining the identical images. So they are mostly asymmetrical, but few are radially symmetrical. It's not mentioned in NCERT book, but generally they focused on asymmetrical nature of the sponges mostly. So the level of organization in the animals is the cellular level of organization. As I mentioned earlier, no animal is unicellular. All animals are multicellular animals. Their body is constructed with many cells. Though their body is made up of with many cells, you know well, the cells are loose aggregates. And the level of organization, which is very primitive level of organization, is cellular level. These are the primitive animals, first animals, earliest animals these are. The earliest metazoans are poriferans, sponges. Moving on, the unique character of the feature of this poriferans is water transport or canal system. It is a unique feature of the phylum Porifera. You can see the different animals over there, the poriferans with the porous nature is given the sponges, different types of sponges with a porous nature. And this unique feature, the canal system, is with the ostia. These are the inhalent apertures through which the water enters. Here we are showing the mark over here, which indicating the water enters through these ostia. Is there minute pores, several minute pores are present on the surface of the body. And this water, it enters into the cavity called as spongocele. Cele meaning cavity. It is a cavity of the sponges. Water enters through ostia and enters into the spongocele, and it leaves out through exhalent aperture called as osculum. Water enters through ostia, leaves out through osculum. In this picture also you are observing only the osculums are very few, but ostia are numerous ostia are present, but osculum is very few osculums are present. It is a unique property, feature of this poriferans. For what sake this canal system, in what way this canal system is helpful? This canal system is helpful in gathering of the food gathering, and is also helpful for respiratory exchange, the exchange of gases, intake of oxygen and leaving out of carbon dioxide, and also the removal of the waste materials. In this way, the canal system or water transport system is helpful. Moving on to the very next important point, that is choanocytes. I am showing here the picture how they look, that the collar cells, they have a collar-like structure, and they have here, you are observing a flagella-like structure over here. This collar cells, the choanocytes, it surrounds the spongocele and canals. It is one of the previous NEET question. The students face this question as such like this: the choanocytes or collar cells lines, or the spongocele or canals are lined by which cells? Their choanocytes or the collar cells. They line the spongocele or canals. It's a very important point. Choanocytes or the collar cells lines the spongocele. What is spongocele? It is a cavity. It is a cavity of sponges. And this, coming to the next point about its digestion, it is an intracellular digestion. The digestion occurs inside the cells. Based on your previous knowledge, I hope you are remembering that with the introductory part of this animal kingdom. In sponges, though they are multicellular, the cells are not organized to form as a tissues, and you know well here these cells show functional isolation. The cells are functionally isolated. They perform different cells perform different functions. The body of poriferans is supported by the skeleton made up of its spicules or spongin fibers. These are the protein fibers and the spicules. The spicules and the spongin fibers will constitute the skeleton, and you know pretty well the skeleton will give support and protection to the organism. Moving on, their sexuality, their hermaphrodites. Hermaphrodites are nothing but the animals in which the sexes are united. The sexes are not separate, the sexes are united. They are called as hermaphrodites or bisexual or monoecious. These are all the terminology we can use, the synonymous like hermaphrodites, bisexual or monoecious. The sexes are not separate. Poriferan, the sponges can able to produce both the male and the female gametes. The male related and the female related, the things are present in the same organism. And it reproduces both asexually and sexually. Asexual reproduction is by fragmentation. If a sponge is broken down into pieces, each piece will develop into a new sponge. Fragmentation is an asexual mode of reproduction. Sexually, it reproduces by producing gametes. It produces the male gametes and the female gametes, where the male and the female gametes will fuse inside the body of a sponge. So, we are referring it as a internal fertilization. So, the fertilization is internal because the gametes are uniting inside the body. And the development, during the development of a sponge, it includes a larval stage which is differing from the adult. And the development is indirect. It passes through the larval stage. They have several larval stages in different sponges, and these are different, quite different from the adult. So, such type of words you need to be in a memorized state, that in which the development is direct, in which the development is indirect, and in whom the fertilization is what the type of fertilization is. It is it external fertilization or internal fertilization? So, you want to concentrate a little bit on this. So, the development is indirect development because it including the larval stage, and the fertilization is internal. And moving on to the examples, the pictures which you are seeing over there, those are the examples of poriferan, the cycon, the scypha. The cycon, the scypha, the spongilla, freshwater sponge. We said that generally majority of them are marine, but few are freshwater. Among this, the spongilla is a freshwater sponge. Euspongia, bath sponge here. So, these are the good examples. You need to learn the examples, and most important, specifying the points like what I told, the type of fertilization, the type of development it is showing, and the unique feature of the phylum Porifera. As I said earlier, you should focus on a few points, and if you go in a comparison way, this chapter will become very easy for you to, because though it's a memory based, you need to memorize all the things related with one phyla and with other phyla, but yet if you focus on a few points, it will become a little bit easy for you comparatively. The next phyla, Coelenterata or Cnidaria. The coelenterates are cnidarians. Why these two names? What it refers actually? Before going to discuss about it, let us discuss about its habit and habitat, where they live and how would be these animals. These are aquatic. Actually, these are aquatic organisms. They live in the water, and they are mostly marine. Here the same thing like poriferans. They also we mentioned there, they're generally marine, means a few are freshwater. In the same sense here also, mostly they're marine, so obviously there are a few of freshwaters. And they're sessile, money, they're attached to the substratum. They cannot, some are sessile, like the picture which I drawn over here, the hydra, they are sessile. Attached to the substratum they are attached. And some are free swimming, like jellyfishes, some they'll swim freely in the water. And these cnidarians, you know well, they are the earliest eumetazoans, I mean the true animals. Kingdom Animalia is also referred as metazoans, you know well. So, the first metazoans are the poriferans. Though their body is constructed with many cells, there we mentioned the cells are not organized to form into tissues, but the tissues are seen for the first time in the phylum Cnidaria. So, they exhibit tissue level of organization and they are diploblastic. Their body is constructed, made up of with two germinal layers, that is outer ectoderm and inner endoderm, and in between these two layers there is an undifferentiated layer, that's what we discussed as mesoglia. Though here it's not mentioning, but you should take that points which mentioned earlier. So they are diploblastic organisms with tissue level of organization, and as tissues are seen for the first time in these animals, these are the true metazoans, or you can call it as a eumetazoans. First eumetazoans. Eumane true. And coming to the symmetry, what type of symmetry they exhibit? Radial symmetry. The animals exhibit radial symmetry. You know well what is radial symmetry. Any plane passing through the center of the animal, it gives rise to the identical halves. So, I am concluding all the points which are the basic phenomena. These are, they exhibit tissue level of organization, diploblastic with radial symmetry. As I said, all are marine, not all, a majority of them are marine, but all are aquatic. All are aquatic, but mostly marine. So, here also some are freshwater farms. And as I said, why we are calling it as a Cnidaria, phylum Cnidaria, based on what it termed as. The phylum Cnidaria is due to the presence of the cells cnidoblasts or cnidocytes present on the tentacles. These cnidoblasts or cnidocytes are the cells, these are the stinging cells, the stings. Because of the presence of these cells, this phylum is named as Cnidaria. And what for, ma'am, these cnidarians, why they used to have this cnidocytes? And these cnidocytes present over the tentacles, they have a fluid-filled capsule called as nematocyst. They have a fluid-filled capsule. This, because of this presence of Cnidaria possessing this cnidoblasts or cnidocyte cells, the predators or other animals, they're scared about this. They're not going to nearby. I mean, they don't want to come nearby to this cnidarians because they know well about what these cells will do. So, they discharge some thread-like structure, and that is helpful for anchorage to stick or to fix to some substratum or something. Defense mechanism, as I said now, these stinging cells will protect the cnidarians. No one is dare enough to approach it. And they are also helpful to capture the prey. They'll throw that thread-like structure and that will sting to the prey and they'll grab it. So, overall, the cnidoblast cells are the cnidocytes. Blasts or cytes, you know, that refers to the cells. These cnidoblasts or cnidocyte cells are helpful for anchorage, capture of the prey and the defensive mechanism. This is one of the unique property of this cnidarians. And why it's called as Coelenterata. The name, I told you the phylum's name, Coelenterata or Cnidaria. Why Cnidaria? We justified now. Coming to why it is Coelenterata, you are observing in the picture they have a cavity. It's named by Coelenteron. The cavity's name is Coelenteron, or it's also called as a gastrovascular cavity. Why? I'll justify it. This cavity, the Coelenteron, because of the presence of this Coelenteron, it's formerly called as coelenterates. Now, in majority of the times, we refer them as cnidarians rather than coelenterates. But anyway, in our NCERT book, most of the times they said like coelenterata, so you can consider this also, no issues. So, coelenteron are this cavity. It opens to the outside through a single opening. You know, we can call it as a sac-like gut. And this opening, it serves as mouth during ingestion and anus during egestion. And why it's called as gastrovascular cavity? The food gets digested over here and also distributes from over here. So gastro related to the digestion, vascular related to the circulation. So coelenteron is otherwise called as gastrovascular cavity, and as it have only single opening, that is mouth, and this mouth is located on hypostome. I'll write here this hypostome here. Mouth is located on hypostome. You know well, stomium mane, it related to the mouth. So this is regarding its cavity and all. Moving on to the next point regarding its digestion, it shows both extracellular digestion and intracellular digestion. Students will get a doubt of basic thing, ma'am, when we are supposed to call it as extracellular and when it's called to be intracellular. If digestion occurs outside the cell, money in the cavity like coelenteron, it's called as a extracellular digestion. Extra, outside, cellular, cell. The digestion is happening outside the cell. If it happens in the coelenteron, it is extracellular. And even the food is also digested in the cells lining the gut, I mean endodermal cells. If the food gets digested in the cells lining this gut, I mean lining this cavity, it's called as intracellular digestion. So it takes both types of digestion happens here, extracellular and intracellular. Moving on to the next point, in some of the cnidarians, their skeleton is made up of with calcium carbonate, the calcareous skeleton. Example for this like corals, they're semi-precious stones. The skeleton of cnidarians we are using in the ornaments, the semi-precious stone, the corals. Coming to the very unique feature of the phylum Cnidaria, they exhibit dimorphism, di-2 morphoforms. The cnidarians exhibit in two forms. One is the polyp stage, other is the medusa stage. What is this polyp and medusa? You can see the difference between these two pictures. The polyp, the good example like Hydra and Adamsia, these are polyp forms. In them, the body is cylindrical shaped body and they are sessile. As I draw the line underneath, it represents their sessile attached to the substratum. So they have cylindrical shaped body, sessile, and you are observing the mouth is oriented upwards. In them, the mouth orientation is upwards over here. And coming to the case of here, one more thing, the medusoid forms. In the medusoid forms, you are observing these are umbrella shaped structures and these are free swimming forms. And in them, the mouth is orienting downwards. This is cylindrical body, this is umbrella shape, mouth is oriented upwards, here the mouth is oriented downwards. These are sessile and thus these are free swimming. So polyp and medusa. In some of the cnidarians in whom they possess these two forms, they exhibit the alternation of generation. We said like the polyp stages like Hydra and Adamsia, the medusoid forms like Aurelia and jellyfishes. In jellyfishes, the body is umbrella shaped and they are free swimming. And moving on to this very important point here, So many important points you'll come across in this phyla. So concentrate on those points. I'll make up ones later. At last, in the forms of cnidarians, in some of the cnidarians, whom they possess this polyp and this medusoid forms, they show alternation of generation, or we can call it as metagenesis. What's this alternation of generation? As I said that they possess the two forms of polyp and medusa. The polyp form undergo asexual reproduction and forms the medusoid form. Medusoid form undergoes sexual reproduction and forms the polyp form. There is an alternation of generation. Asexual followed by sexual and sexual followed by asexual form. The good example for this is Obelia. Obelia is a good example for this alternation of generation or metagenesis. Examples, the Physalia, Portuguese man of war. The shape of this looks like a warship of Portuguese. Physalia, Portuguese man of war. Adamsia, the sea anemone, which is also the polypoid form. Pennatula, sea pen, pen. Okay, already I told you mostly they are marine. Gorgania, sea fan, it's like looks like a hand fan. And Meandrina, it is a brain coral. As I said, the corals are the semi-precious stones, as I said. And what to concentrate in this phylum Porifera? We said like some unique features. They possess the cnidoblasts or cnidocyte cells, which helpful for them in the defensive and other purposes. And they have the dimorphic nature of polyp and medusoid forms, and some they exhibit alternation of generation like Obelia. And moreover, I told you already, focus on the examples. You need to mug up or you need to go through several times in order to know both the scientific names and also the common names. The next phyla, Ctenophora. Ctenophorans are commonly called as sea walnuts or comb jellies. The name itself indicating they are marine forms, sea walnuts or comb jellies. They are exclusively marine. The previous two phyla, mostly marine they are, Poriferans and Cnidarians, they are mostly marine. But Ctenophorans are exclusively marine. No freshwater form is represented from this phyla. You can see some of the comparative features between the phylum Cnidaria and the Ctenophora. Both are diploblastic, show tissue level of organization, radially symmetrical animals. And even the digestion in both of them also both extracellular and intracellular digestion. So you can see the comparative ones like in both the Cnidarians and the Ctenophora members, they are of radial, symmetrical, diploblastic organisms with tissue level of organization. And even the digestion is both extracellular and intracellular digestion. And where it differs, in which phenomenon it differs with the Cnidarians. In Cnidarians, they have cnidoblasts or cnidocyte cells, but these don't have those cnidoblast cells or cnidocyte cells. Cnidoblasts or cnidocyte cells are absent in the members of this Ctenophora. And in the Ctenophora members, they have some special feature. They have external 8 rows of ciliated comb plates, and this is the characteristic feature. You can see in the picture, they have 8 rows of comb-like plates are present. They are externally located ciliated structures. As they possess cilia, obviously they are helpful for locomotion. And moving on to the next, a very interesting characteristic feature of Ctenophora members, bioluminescence. You know, bio, life, living organisms, luminescence, light. The emission of light from the living organisms is called as bioluminescence. The bioluminescence is the characteristic feature of the phylum Cnidaria. They emit the light from their body. For what sake they'll emit the light for the body? In order to attract their prey, in order to attract their mates, and also in order to make their predators stunned. If suddenly a light glows, obviously will be stunned. So bioluminescence is the property, a characteristic feature or a special important phenomenon of the phylum Ctenophora members. Coming to their reproductive part. They're bisexual. Sexes are not separate, the sexes are united. And as you learned the same phenomenon in Cnidaria members, they're monoecious or hermaphrodites. Sexes are not separate, they're united. So we are using 3 to 4 terminology in order to refer it. And the reproduction, important point, as I said, like exclusively marine. In the same way, the reproduction in these animals is only through sexual reproduction. They reproduce only sexually. In Cnidarians, we come across both asexual and sexual, but here only sexual reproduction. And the fertilization is external. The gametes fuse outside the body in their media, and the development is indirect development. So they pass through the larval stage. Development is indirect, fertilization is external. Coming to the examples of this, Pleurobrachia and Ctenoplana. Pleurobrachia and Ctenoplana are the good example for the members of Ctenophora. So you want to focus on the points like how many rows of the comb-like plates are present. 8 rows, ciliated, meant for what? A cilia, obviously for locomotion. And what is a special feature? Bioluminescence, exclusively marine, and reproduce only through sexual reproduction. Next phyla, Platyhelminthes. Platy money, flat. Helminthes means worms. These are flat worms. Their body is dorso-ventrally flattened body. Hence, they're called as flat worms. Their body looks like a ribbon or like a leaf. It's flattened body. And most of the Platyhelminthes members are endoparasite. Endo, inside. They live parasitic, they lead a parasitic life inside the body of hosts. And majority of them are the parasites on human beings. But few are the free living forms are also there, like planarians, which are the free living members of this Platyhelminthes. And coming to their descriptive phenomenon, these are the first triploblastic animals. That the triploblastic means, their body is made up of 3 germinal layers. They possess the 3 germinal layers, the outer ectoderm, the middle mesoderm, inner endoderm. In the previous classes, I told you the mesoderm in triploblastic animals is responsible for the complexity of organisms. So that triploblastic, bilaterally symmetrical, and they show what type of coelom, acoelomates, no coelom. Coelom is absent in them. And the level of organization is organ level of organization. You don't need to get confused over here, no need of confusion, because already all this terminology you come across in the case of the first topics itself. And coming to some of the parasites like Taenia solium, the tapeworm, in them, in order to lead a parasitic mode of life, they have hooks and suckers. They have hooks and suckers in order to attach to the host body for adhesion phenomenon, to acts like a hold fast. These are hold fast organs to lead an endoparasitic life. If we talk about the Taenia solium, the tapeworm, it is a gut parasite, it lives in the gut. Whenever the gut shows this contractions, the peristaltic movements, is a chance of expelling this parasite outside along with the fecal matter. In order to avoid it, in order to prevent that expelling of this worm outside, they need to attach, they'll attach to the wall of the gut inside with the help of this hooks and suckers. Next phyla, Aschelminthes, the pseudocoelomate phyla. In some of the books, it's also referred as a nematoda, but according to the NCRT concept, is Aschelminthes, the pseudocoelomate phyla they are. And coming to this, they're commonly called as roundworms. Why? If you do the cross-sectional view, they looks round in shape. The cross-sectional view of these worms is round, hence they're called as roundworms. And they're free living organisms. You can see a diversity in their living. Some are free living, aquatic, terrestrial, and some are parasite on plants and animals. There are different modes of living as seen in this phyla, Aschelminthes phyla. As I said, all the pseudocoelomate phyla are incorporated in this. And they are triploblastic, bilaterally symmetrical, and pseudocoelomates. Already you learnt about what are pseudocoelomates. Here in these animals, the mesoderm is scattered as a pouches in between ectoderm and endoderm. Though they have a cavity, but is not a true cavity, is not lined by the mesoderm and is not formed by the mesoderm. Hence, we are calling it as a FAL, pseudocoelomate. And the level of organization is the organ system level of organization. In Platyhelminthes, organ level, and this is here organ system level of organization. Coming to the gut part, they show complete gut. We can say here it is a tube within a tube organization, a tube within a tube organization here. A complete gut is seen, a tube like gut where one opening for ingestion, another opening for egestion. They have two openings. And previously whatever we discussed in the phyla like Cnidaria and in the members of Platyhelminthes, they have a sac-like gut with a single opening, incomplete gut, but here is a complete gut. And the gut is non-muscular, but pharynx part is muscular. It is a highly developed muscular pharynx is seen. Generally the body wall is muscular, but alimentary canal is non-muscular, but pharynx is highly developed muscular part here. That's a phenomenon that you want to focus on. Coming to their excretory system, excretory one here, the excretory system includes the tube which eliminates the waste through the pore called as excretory pore. Excretory tubes and excretory pore play a role in the elimination of waste. Moving on to their reproductive part, here it is, they are dioecious. Dioecious money, sexes are separate. They are male and the female worms. Moreover, they exhibit the sexual dimorphism. You can see in the pictures over there, the male and the female show clear demarcation. Male is short with a curved posterior end, whereas the female is long with a straight posterior end. They are sexually dimorphic, sexually two forms, male and the female. Dioecious, males are shorter with a posterior curved end and curved tail, and whereas in the females you can see the posterior end is straight and they are of long comparatively with the males, clear demarcation. And coming to their fertilization, they show internal fertilization. They exhibit internal fertilization, and moreover the development may be either direct development or indirect development, because some of the Aschelminthes members are viviparous. They can give birth to the young one. No need to go with that much of deviation, but in order to justify that, in some the development is direct. They don't pass through the larval stages, but in some it is indirect development. Examples: Ascaris, the common roundworm, and Eucraria, the filarial worm, which you will come across the type study in another chapter, the structural organization in animals. And the Eucraria is the filarial worm, where you will come across the type study in human health and diseases chapter. And Ankylostoma is commonly called as a hookworm. Ascaris, the common roundworm, Eucraria, the filarial worm, Ankylostoma, the hookworm. So what need to stress the points here in the aspect of neat point of view here. A complete gut, pseudocoelomates, a tube within a tube organization, and moreover sexually dimorphic structures, male and the female is clearly demarcated from one with other.